In this video, we're going to be looking at the process of protein synthesis, which we can actually break down into two kind of smaller steps known as transcription and translation. Remember that the word synthesis means to make. So this lesson is going to talk about how cells make proteins. So remember that the process of protein synthesis can be broken down into these kind of two smaller sub-steps. The first one is transcription, and the second one is known as translation. And you can see in this image here that during transcription, a molecule of DNA gets converted into a molecule of RNA. And more specifically, we're going to be talking about a type of RNA called mRNA. And that M stands for messenger. During translation, the RNA is going to be kind of converted into a protein, or at least it's going to provide the instructions needed in order to make a protein happen. This slide shows a kind of a still shot from a video. I can't play the video in this program here, but if you access the original slideshow on Canvas, you should be able to click on the play button, which will show up in the front here like this, and you can watch this video that kind of helps to explain the overview of protein synthesis. Now notice you might have to put this into present view in order to be able to watch that video. So present will be kind of up in the upper right hand corner. So you'll click on present. It'll show up like a presentation, and then you should be able to hit that play button to actually watch the video. Recall that genes are just sections of DNA and chromosomes that code for proteins. So you can kind of think of the genes as being the recipes for how to make proteins. In fact, one gene provides the information needed to make one protein. So your genetics or your genes determine what proteins your body makes. Remember that your genes are just these little sections of DNA that are stored in your cells in the form of these chromosomes here. Now you can see over here, this is something called a karyotype. And this is showing all of the chromosomes in a particular individual. And you can kind of see these bands on the chromosomes here. Now each one of those bands actually can hold hundreds to thousands of genes. So each band is not its own gene. The genes are even smaller than that. But genes can be made up of a really long strand of DNA in order to provide that information needed so your body can make a protein. So let's review a little bit about where DNA is located, what organelles we need in order for protein synthesis to occur, and where the organelles for protein synthesis are located. So remember that in eukaryotes, one kind of defining feature of eukaryote is a cell that has a nucleus. All right, and this is a membrane-bound nucleus. So it's a nucleus surrounded by a membrane, kind of like the cell membrane. So the answer to this question is that DNA is located in this membrane-bound nucleus. Now, the organelle that's necessary for protein synthesis to occur are the ribosomes. And remember that the ribosomes are going to be located in the cytoplasm, or you could say the cytosol. Now, they may be free-floating in the cytoplasm, or they may be attached to the rough ER, which is the rough endoplasmic reticulum. Even if they're attached to the rough ER, they're still located in the cytoplasm. So the key thing is that we have to get the information from the nucleus to the cytoplasm. And that's where our RNA is going to come into play. So as we outlined on the last slide, we need DNA to make proteins, but DNA is stuck in the nucleus and the ribosomes are stuck in the cytoplasm. So how do we get the information from the nucleus to the cytoplasm? Because DNA is too big to leave the nucleus. So how do we get this information stored in DNA from the nucleus out into the cytoplasm? And the answer to that conundrum is this molecule known as mRNA. Now, mRNA is smaller than DNA. And it's smaller in two ways. So it is shorter than DNA. It's also only a single strand. And because of its size, it is able to fit through the pores in the nuclear membrane. So mRNA is made in the nucleus, but then it is able to leave the nucleus through these little nuclear pores, which are just little openings in the nuclear membrane. And then it can go out into the cytoplasm to find a ribosome. During the first part of protein synthesis, known as transcription, a molecule of mRNA is made from a molecule of DNA. So a gene, which is, of course, a segment of DNA, gets converted into a molecule of mRNA. Now, this conversion happens through complementary base pairing, which is similar to what you learned when you learned about DNA replication. Remember that during DNA replication, you have these two strands of DNA that are complementary to each other. So you've got your G, C, A, and T. And then the complementary strand, we know that C always pairs with G pairs with C pairs with G, A pairs with T, and T pairs with A. Now during DNA replication, these two strands are going to separate. And then each of the original strands becomes like a template for a new strand that's going to be made. And the new strand is just complementary to the old strand. All right, and I'm out of room, but you should remember that from when we did DNA replication. Now, in transcription, something kind of similar happens, but instead of the new strand being DNA, instead that new strand is going to be RNA. And the two strands of DNA are not separating completely, and they're not separating forever. It's actually just a temporary separation of the two strands of DNA, and it's only a portion that gets separated. So what happens is that you have this double helix of DNA, and what happens is that just a section of it is going to open up. And this is the section that is the gene that is being expressed. So this DNA kind of opens up. And then we have enzymes known as RNA polymerase that is going to come in and basically make a strand of mRNA using the DNA as a template. So this area in yellow here is going to be our mRNA. Now, another key difference is that RNA is made up of nucleotides, just like DNA is. So remember that nucleotides are made up of a 5-carbon sugar, a phosphate group, and a base. Now, the 5-carbon sugar in DNA is known as deoxyribose, but in RNA, it's known as ribose. So it's ribose in RNA, and it's deoxyribose in DNA. The other key difference has to do with the types of bases that are present. So in DNA, we have what we call, or what we kind of remember as GCAT, where guanine always pairs with cytosine, and adenine always pairs with thymine. Now, mRNA does not have thymine. Instead, it has what I like to call GCow. So instead of thymine, it has this molecule known as uracil. So no thymine, instead it has uracil. And the base pairing still applies, where G still pairs with C, and A pairs, in this case, with U instead of T. So if we have a molecule of DNA with the bases of GCAT, the corresponding RNA, G is still going to pair with C, and C is going to pair with G. Now, if this were DNA over here, which is not, instead it's RNA, there would be a T here. But remember, we don't have T, so instead that A is going to pair with a uracil. Now, the T is still going to pair up with A because we still have adenine. So really the only difference is going to be that U pairs up with A. With the U is in RNA, and it's going to pair up with the adenine in DNA. So to recap, some differences between DNA and RNA. So first of all, mRNA stands for messenger ribonucleic acid. So the M stands for messenger, and then ribonucleic acid. And we can break down this word a little bit. So ribo refers to the ribose sugar, which is just a 5-carbon sugar. And so it's going to look kind of like a little ring, kind of like a little pentagon. The nucleic acid part of it tells you that it is a nucleic acid, which remember is one of our macromolecules. And remember that macromolecules like DNA and RNA, like our nucleic acids, are made up of monomers, which are the nitrogenous bases. And then remember, the big difference between RNA and DNA is this base known as uracil. So in DNA, this base would be thymine. But in RNA, it's called uracil. The other main difference, as you can see in the image over here, is that DNA is double-stranded, whereas RNA is single-stranded. So this makes RNA thinner, and it's one reason why it can leave the nucleus and go out into the cytoplasm, is because it can fit through those small pores in the nuclear membrane. So let's take a more in-depth look at what happens during transcription. So we know that RNA is made from DNA, and we know that it occurs in the nucleus because that is where our DNA is stuck. It is stuck in the nucleus of eukaryotic cells. So this image here is showing you how transcription happens in eukaryotes. So these two strands right here are showing you the double-stranded DNA. So this is the kind of like the base molecule. Remember that this is the gene, all right? And this gene is going to be providing the instructions ultimately for how to make a protein. But before we can make a protein, we first have to make mRNA. So what happens is that the gene is actually down here, kind of like downstream of this area that we call the promoter. And what happens in this promoter region is that we have transcription factors, which are proteins, and we have enzymes, and we have some other stuff that comes together and it binds to the DNA at this promoter region. And you can see that we call the promoter region, we kind of call it this TATA box. And it's because it's made up of a whole bunch of thymines and adenines that are repeating in this area. And so there's some sort of signal to the transcription factors that is where they should initially bind to and then start the transcription process. And as you can see here, we've got our double-stranded DNA, and we have just one small area of the DNA that unwinds. So we're not completely separating the two strands of DNA like we did during DNA replication. Instead, we're just opening up just the gene and a little bit of the area around the gene. So we're really just exposing the gene that we want to transcribe. And we call this the transcription bubble because it's only this area that's going to be transcribed into RNA. So the two strands of DNA separate and the DNA bases become exposed. Now only one strand is going to get transcribed into forming this mRNA molecule. And so there is an enzyme, RNA polymerase, and if you look at the word RNA polymerase, it tells you what it does. Remember that ACE, that ending of ASE, means that it's an enzyme. And it's an enzyme that makes a polymer of RNA. So it's taking those nucleotide monomers and putting them together in order to form a polymer, which is a longer, bigger strand of RNA. Once the gene is transcribed completely, the mRNA is going to detach from the area, and the two strands of the DNA are actually going to come back together as if nothing happened. So we're not damaging or destroying DNA in the process, we're just temporarily opening it up so that we can make this molecule of mRNA, and then it goes back together until that gene needs to be transcribed again. Eukaryotic DNA actually contains, or the genes contain segments of DNA that are needed to make the protein, but there are also some kind of areas of DNA that are not needed to make the protein. And in fact, they kind of get in the way when trying to make a protein. So exons are just these areas of genetic sequences or DNA that we need in order to code for the protein. So on the picture down here, we're looking at these blue segments here. Now the orange segments are indicating introns, and introns are just sections of DNA that actually get in the way. They don't code for a protein. I don't know if we know really what they do. They might have something to do with which genes are expressed, or we might just not know. But essentially, before we go into our next step of translation, we have to get rid of these areas of genetic information that need to not be there. Now, remember that in DNA, the mRNA is being made from the DNA. So these nucleotides, so let's say we've got some C's and G's and T's and A's and all that in here, they're still getting converted into our mRNA. So what we end up with is a strand of mRNA that has some useful information, but also some information that we need to get rid of before we go into our next step of translation. So after transcription happens, so we can see here that the DNA gets converted into the mRNA. But this mRNA needs to be processed because it contains the introns and the exons. And we have to get rid of those introns because remember, the introns they're not needed to make proteins, and in fact, they're going to get in the way. So what happens during this post-transcriptional processing is that after transcription happens, there are enzymes that go in and they remove these nonsensical areas that are not needed. And then it actually splices the RNA back together. So we end up with this mRNA molecule that has the correct information needed to make a gene, but we've gotten rid of all of the information that we don't need. So we end up with this kind of shortened RNA molecule that only contains the information that we need. Now, in addition to cutting out the introns in order to only have the exons, the ends of the RNA molecules also get some extra nucleotides added to them. So you can kind of see that on this picture here. You get a 5' cap, and so these are just extra nucleotides added to the 5' end of the mRNA molecule. At the other end, you get this thing called a poly A tail, which is just a whole bunch of adenines added on to that end of the mRNA as well. And these added nucleotides primarily serve a protective function. So think about it, this molecule is going to have to go from the nucleus, and it's going to have to squeeze out through those nuclear pores in order to get to the cytoplasm. And along the way, it might bump into things, it might be exposed to some chemicals that could be dangerous to it. And so these extra nucleotides are sacrificial, because if we lose a little bit of information here, it's no big deal because we don't need this information in order to make a protein. Whereas if we lose information from the actual coding part of the mRNA, now we're going to end up with something like a mutation, and we're going to end up with a protein that isn't made properly. Now we're going to take a look at the second part of protein synthesis. So during transcription, we made a molecule of RNA, and more specifically, we made a molecule of mRNA. Now the mRNA, remember the DNA is stuck in the nucleus, but to make a protein, we need an organelle called a ribosome. And so the mRNA is able to carry the information in the DNA in the nucleus out to the ribosome, which is located in the cytoplasm. For the process of translation to occur, we actually need 3 different types of RNA. One type is the mRNA that we just talked about. And remember that this is just the DNA getting converted into mRNA during that process of transcription. Additionally, we need rRNA and tRNA. rRNA is also known as ribosomal RNA, and it actually helps to make up part of the ribosome. And tRNA stands for transfer RNA, and transfer RNA transfers or carries amino acids to the ribosome. So what happens is that the messenger RNA goes to a ribosome, and the ribosome is kind of like the factory that it can read the messenger RNA, and it helps to bring the transfer RNA into the ribosome, and then it takes the amino acid. And as it takes the amino acids, it starts making this chain of amino acids, which is ultimately going to become the protein. So let's take a look at how that happens on the next several slides. Now, messenger RNA is made up of several of these nucleotides, or these bases, that are all bonded together. 3 bases, or 3 nucleotides in RNA, is known as a codon. All right, so this molecule of RNA is made up of these triplets called codons. And each codon, it codes for one amino acid. So you can see we actually have this chart over here that helps us to kind of figure out which codon codes for which amino acid. Now, this chart, you don't have to memorize this chart. You just need to be able to use it as a tool. So just like the periodic table, you don't have to memorize all the individual elements and where they are and how many atoms and stuff they have. Instead, if I give you a chart like this, you need to be able to use it to figure out which codon codes for which amino acid. So let's take a look at this codon here. So this GUC that we see right here. So the first letter is going to be G. So that means that our amino acid is going to be in this row on the chart right here because our first letter is G. Our second letter is U. So that means that our amino acid is going to be in this square right here. And that's because U is our second letter, which means that we can take a look at where these two, the column and the row, overlap. Our third letter, which I kind of messed up, but that's a C right there, is going to be this row right here. So we can find our codon here for GUC, and that translates to our amino acid known as VAL, which is also known as valine. So anytime there is a GUC on an RNA sequence, it's going to code for an amino acid known as valine. Now we can look at this other codon here. So we have CCA. Let's do this one in blue. So in this one, our first letter is our C. So we know that the amino acid is going to be in this row right here. Our second letter is C, so it's going to be in this row right here. So we can look at where the two overlap. So we're narrowing it down. And then our last letter is A, which is the third one down. So you can see CCA codes for PRO, which is the same thing as proline. So now we know, and these are the amino acids over here that I'm writing here. So if we have a sequence of DNA, we can then figure out which, or what the sequence of mRNA is going to be, and then we can figure out what the codons are, and then use this chart to figure out what the amino acid sequence is. So essentially, to kind of break this down, you have DNA that gets converted to mRNA. The ribosome exposes one codon at a time, and each codon gets, or it kind of codes for the location of an amino acid. And a chain of amino acids is what gives you a protein. Now, in addition to this chart coding for the different amino acids that are going to be in a protein, we also have what we call start and stop codons. So the start codon is AUG, and it codes for an amino acid known as methionine. So all proteins start with the amino acid methionine. And the reason why we have to have this start codon is because remember that during our post-transcriptional processing, remember that our molecule of RNA got that poly A tail and that 5' cap added to either end. But these do not need to be translated into a protein. Remember, these are just throwaway nucleotides that are there for protection. And so the ribosome knows to start when it finds the codon of AUG. And so the ribosome is going to come in and expose just that codon there. Now, this translation is going to happen kind of in one direction along the RNA, where the ribosome is now going to expose the next codon and then the next codon. But ultimately, it has to stop, and it has to stop before it gets to the other end of the RNA, because once again, the 5' cap and the poly A tail are not supposed to be translated. And so if the ribosome comes across either UAA, UAG, or UGA, these are known as the stop codons, and it's kind of a signal to the ribosome that it needs to stop the translation process, and it detaches from the RNA, and then the protein is done being made. Once again, you don't have to memorize these codons. Just be able to read the chart and understand how to figure out which codon codes for which amino acid, and be able to know where the start codons are based on what we see in the chart here, and where to stop based on the stop codons here. So recall that the mRNA has a long strand of nucleotides. And these nucleotides, the order of them was determined by the strand of DNA that it was made from. Remember, we've got our guanine, cytosine, adenine, and then instead of thymine, we have uracil. And so we can go ahead and just kind of finish out this strand here by putting some random nucleotides in. Now, remember that 3 mRNA nucleotides is equal to a codon. So, for example, this GCA would be a codon. The UUA would be a second codon. The CCG would be a third codon. And then the A would be the first part of the later codon here. Now, the mRNA meets up with the ribosome, and the ribosome actually exposes one codon at a time. So the ribosome is basically helping to kind of expose just one codon at a time. And exposing that codon helps the tRNA be able to kind of meet up with the mRNA. So what happens is that tRNA, our transfer RNA, it has 3 bases attached to it. And these 3 bases are known as an anticodon. So as our ribosome is exposing this codon here, the tRNA that has the matching or complementary anticodon is going to kind of fit together with this codon like a puzzle piece. So for example, this would be C pairing with G, G pairing with C, and U pairing with A. Now, this tRNA molecule has an amino acid stuck to it. All right, and remember, we can use that chart to figure out which amino acid is stuck to it. So what happens is that the ribosome, so we'll kind of draw like a little dotted line here. So the ribosome comes in, it exposes this codon and allows the tRNA to come in with the matching anticodon. And once that match is made, the ribosome actually takes this amino acid and it holds on to it. I guess I should do that in yellow. Then that ribosome is going to move down the line to expose the next codon. And so another molecule of tRNA is going to come in, and it's got to be complementary to this codon. So in this case, it would be AAU. And this tRNA would have a different amino acid attached to it. So the ribosome, like I said, would move down, and the tRNA would come in, and it would actually drop off its amino acid, and the ribosome would form a bond with the amino acid it got from the first tRNA molecule. So here, I'll label them here as 1 and 2, and 1 and 2. Then the ribosome, is going to move down to expose the next codon, which is going to allow the next tRNA molecule with its anticodon to come in. And it's going to be attached to amino acid number 3. And once it matches, the ribosome is going to take that third amino acid from the tRNA and form a bond with the other amino acids it got from the first 2 codons. And this is going to keep happening until the ribosome hits that stop codon, and then it's going to release the mRNA, and it's going to release this polypeptide up here. So this chain of amino acids that's forming is called a polypeptide. And this is just a chain of amino acids. So you can see in this picture down here, this is showing the mRNA here and the ribosome here. And we've got our tRNA molecules with our anticodons. And we have the ribosome exposing codons one at a time. And you can see how the tRNA molecules are coming into the ribosome. And if the anticodon matches the codon in a complementary way, what happens is that the amino acid gets dropped off at the ribosome. And you can see that the ribosome is going to take that amino acid and form a bond with the last amino acid it had. And so this is going to keep happening until we hit that stop codon further down the messenger RNA molecule. Now remember that the ribosome itself is made up partially of RNA, and it's actually called rRNA, which stands for ribosomal RNA. And it just helps to make up part of the ribosome, and the rest of the ribosome is made up of proteins. And these ribosomes actually have 2 subunits. They have a large subunit and a small subunit. And these subunits can actually come apart and then come back together depending on their usage when they're being used. Now, the RNA and the proteins are made in the nucleolus. Now, remember, the nucleolus is just this region in the nucleus that codes for these ribosomal subunits. And as we've already established, we need the ribosome in order for translation to occur. And it kind of helps to facilitate or kind of play matchmaker between mRNA and tRNA. It helps to kind of bring the whole process together so that the anticodons can match up with the codons. Now let's do a quick review of amino acids. We talked about amino acids way back at the beginning of the semester when we talked about macromolecules. So remember that proteins are one of our macromolecules, and they're one of our polymers. And remember that polymers, so a protein, like we said, it's a macromolecule and it's a polymer. Remember that poly means many. And polymers are just these large molecules that are made up of many monomers bonded together. So remember, mono means one. So amino acids are the individual building blocks that bond together to form this polymer that becomes a protein. Now there are 20 different types of amino acids, but they all have the same basic structure in that they have this central carbon. They have an amino group, which is a nitrogen bonded to 2 hydrogens. They have a carboxyl group, which is a carbon double bonded to an oxygen, and also bonded to an oxygen that's bonded to a hydrogen. they also have this kind of single hydrogen coming off of the central carbon. Now, the key difference between our 20 different types has to do with this kind of what we call the R group or the side chain. So you can look in our chart over here that all of these amino acids have the same basic structure except for the shaded areas here. So some amino acids are really simple. So glycine, instead of having this R, this R is just kind of a placeholder. So instead of having an R there, it would just have a hydrogen atom. But alanine has a carbon bonded to 3 hydrogens. And you can see that valine is even bigger, and it actually has a little bit of branching going on. You can see that this R chain here, or this side chain, it can be a long branch of carbons and hydrogens. You can see that there are also some nitrogens here. Sometimes there are some sulfurs in this side chain. Sometimes these chains form rings. So this R side group here is just a placeholder for all these different possible combinations. And all of these different combinations, they just allow these amino acids to interact with each other in a way that ends up giving the protein a 3-dimensional shape, which we'll talk about that more on a later slide. It's important to note that a polypeptide itself is not necessarily a functioning protein. So a polypeptide is just made up of amino acids bonded together. And so this is what is formed by the ribosome during translation. So each one of these is just an amino acid bonded together to form this polypeptide. And these amino acids are bonded together by specific bonds called peptide bonds. And whenever you're talking about polypeptides and proteins, you're going to see this word peptide show up a lot. So if you see the word peptide, there's a good chance that it has something to do with amino acids and or proteins. Now, a protein itself is typically where this chain of amino acid actually takes on a 3-dimensional shape, meaning that this amino acid is going to start folding around itself. So it's not just going to be a long strand of amino acids. It might be kind of a big glob, and there are kind of different shapes associated with that. So there are actually what we call 4 levels of protein structure. And we're going to talk about those on the next several slides. All right, so the polypeptide that was made during translation is actually the primary structure. So this is just that chain of amino acids bonded together to form a polypeptide. In the secondary level of structure, what happens is that you have these amino acids here that are bonded together in this polypeptide. But remember that these amino acids, even though they have the same basic structure to them, they all have this different side chain sticking off of them. And remember, we just kind of use this R as a placeholder, but sometimes these side chains are just like a hydrogen. Sometimes they're these kind of bigger molecules. Sometimes they're these long chains of carbons. So they're taking on all these different shapes and sizes, and some of them are hydrophobic, some are hydrophilic, some are polar, some are nonpolar. So there's a lot going on with these side chains. And what happens is that sometimes these side chains are attracted to each other, and other times they are repelled by each other. And because of these attractions and repulsions, what we end up with is this shape that starts to form. And so the secondary structure are these amino acids that start folding into either these what we call pleated sheets, and these are called beta pleated sheets, and that's the Greek letter for beta. Or they form these these spirals called alpha helices. And so that's just these amino acids starting to fold into these different shapes. The tertiary level of protein structure is where these alpha helices and beta pleated sheets start to interact with each other, and they start to fold and bend around each other. So you can see here we have these beta pleated sheets, and we have these helices, and those side chains are still interacting with each other, but now they're causing the helices and the pleated sheets to now kind of bend and fold around each other. Now, in some proteins, the tertiary structure is all that it needs in order to be a functioning protein. So some proteins stop at the tertiary structure level, but some of them go into what we call the quaternary structure, which we'll talk about in the next slide. So as I mentioned, the quaternary structure is not present in all proteins, but it is present or required in some proteins. And what happens is you end up with multiple polypeptides that actually go through their secondary and tertiary structures, and then they end up combining together. So as we see in this picture here, we actually have two different polypeptides that have formed their alpha helices and their beta pleated sheets. They've folded into their tertiary structures, and now they're connected together to make a quaternary structure. And in this image, we're only looking at two, but there are some proteins that have even more of these polypeptides bonded together. For example, hemoglobin is a protein in your blood that carries oxygen. And hemoglobin is actually made up of 4 of these polypeptides bonded together. So it's a pretty big protein because it's got these 4 polypeptide subunits that are all kind of bent around themselves and bonded together to form this functional protein. So this slide is just showing you an overview of those 4 levels of protein structure. So as you can see, we start out with our polypeptide sequence here. So this is just our chain of amino acids forming a polypeptide. Those side chains interact with each other to make the beta pleated sheets and the alpha helices. They then bend around each other to form the tertiary protein structure. And remember, some proteins stop at this level and they're functional and they're good to go. But other proteins might need to go to the quaternary structure, meaning that they need multiple polypeptides bonded together in order to function as a protein. So far in this lesson, we have primarily been focusing on eukaryotes. So this course primarily focuses on humans, and humans are eukaryotes. But it is important to understand that there is a difference between protein synthesis in eukaryotes and protein synthesis in prokaryotes. And so here's a little chart that kind of shows us some of the differences. So first of all, remember that in eukaryotes, we have the DNA stuck in the nucleus. But in prokaryotes, remember prokaryotes do not have a membrane-bound nucleus. So transcription and translation are both happening in the cytoplasm. Eukaryotes typically have these linear chromosomes, and we typically have multiple chromosomes, whereas prokaryotes have one single circular chromosome that holds the entire genome of the prokaryote. Remember that in eukaryotes, our chromosomes are kind of this condensed form of chromatin. And chromatin is made up of DNA wrapped around these proteins called histones. So in order for our DNA to be kind of unwound and opened up for transcription to occur, there has to be some extra proteins and enzymes involved in helping to open up that DNA from this highly organized structure. But prokaryotes have a much smaller genome, and they only have this single circular chromosome. They don't have histones, and so it's actually somewhat easier to get access to the genes in order for transcription to occur. In eukaryotes, remember that we had to have that post-transcriptional processing because of really two reasons. So one reason was we needed to get rid of those introns, which are not needed in order to make a protein. Remember that we also needed the poly A tail and the 5' cap. And remember that those were to help protect the mRNA as it was leaving the nucleus and going out into the cytoplasm. But in prokaryotes, we don't have that problem. We don't have the nuclear membrane. The mRNA doesn't have to go very far in order to find a ribosome. So we don't need that post-transcriptional processing, partially because we don't have, prokaryotes don't have introns to begin with, so we don't have to get rid of them. And we don't need the poly A tail and 5' cap because they don't have to travel anywhere. The mRNA doesn't have to travel anywhere to find a ribosome. So now let's talk about this issue with mutations. So mutations occur when DNA is different, or it's changed in some way from what it originally should be. So for example, if the DNA sequence is the G, C, C, T, A, G, C, etc., if all of a sudden the C gets converted to an A, now we have a mutation because the base that is supposed to be in this spot here has changed from a cytosine to an adenine. Now, this can happen for a variety of reasons. First of all, DNA gets replicated a lot, and it's just like if you're trying to maybe type up something from an original, and as you're typing, you hit the wrong key. So you just have a typo. So sometimes during DNA replication, or even during transcription, sometimes the wrong nucleotide just gets put in the wrong spot. Environmental factors can also damage your DNA. So things like exposure to radiation. So for example, UV radiation from the sun can damage the DNA in your skin, which leads to skin cancer. Or you could be exposed to certain chemicals. For example, some of the chemicals in cigarette smoke can lead to damage in the DNA in the lungs, which can lead to lung cancer. Now, if DNA changes, it means that our mRNA is also going to change, right? Because the mRNA is made based on the DNA template. So remember, G pairs with C, C pairs with G. And in our original, we would have a G here, and then the T pairs with A with U, G with C, and C with G. But if our DNA mutates before mRNA is made, now we're going to have the wrong mRNA nucleotide in that area. So we're going to have a U instead of a G. Now remember that the mRNA is what the ribosome uses to make a protein. So now we've changed the code, or we've changed the recipe for how the ribosome is going to make this protein. So ultimately, we're going to end up with a different protein that might be the wrong protein. But the good news is, if the DNA changes and the RNA changes, that doesn't always mean that the protein is going to change. And that's because if we look at our chart over here, so this is our codon chart for our amino acids, and you'll notice that multiple codons might end up with the same amino acid. So depending on where the mutation occurs and which nucleotide is involved and which nucleotide gets changed to which other nucleotide, sometimes it doesn't end up changing the end result of the protein. So for example, let's take a look at what we did up here. So our original codon was GCC. Oh, sorry, just kidding. Our original codon was CGG, and our new codon with the mutation would be CGU. So let's see which proteins are made, which amino acids are coded for by these codons. All right, so C, G, G, codes for arginine, whereas CGU, ooh, surprise, it also codes for arginine. So in this example, the mutation didn't matter because we're still going to end up with this amino acid in that spot in our polypeptide. So no big deal. Now, if it were to, let's say instead of changing this third letter, we change the first letter. So now let's say that G gets changed to a C, which changes our mRNA, the first nucleotide, to a G. So now we've got GGU, and GGU, let's see, so GGU, that codes for glycine. So now we've got a problem because now instead of arginine, we have glycine in that spot, and now we have a protein that doesn't have the right polypeptide chain. It's got the wrong amino acid in the wrong spot. And that means that the secondary and tertiary structures of this polypeptide are not going to be correct because glycine has a different side chain than arginine does. And in doing so, it's going to make our protein fold in a different way, and it's potentially going to make our protein not function the way that it's supposed to. All right, so not all mutations cause a change in the protein. It really depends on which nucleotide is affected, the location of the nucleotide, and which codons code for which amino acids. Now, let's say that our mutation did change the protein that was made. Now, that's not always bad, but we'll start out by talking about some of the bad things and then move on to maybe talking about how sometimes it can actually be a good thing. But let's start out with the bad. All right, so if this mutation causes the protein to be made in a way that makes it lose function, or maybe it's a protein that regulates the cell cycle, and so now the cell cycle is not regulated properly, and it can lead to problems, especially things like cancer, and then most genetic diseases are based on a genetic mutation that leads to a protein not being made properly, and other disorders as well. All right, so these are going to be the bad possible outcomes that can lead to a lowered quality of life or even death. But remember how we said that some mutations are totally neutral because of that redundancy in the amino acid code. So if the change of the mutation does not change which amino acid is located in which area, then it doesn't matter. And we call this a neutral or a silent mutation because you'll never know that you have it because it really doesn't matter. If you end up with the same amino acid code, even with the mutation, then it doesn't matter. It's kind of like with, let's say you've got a recipe for cupcakes, and in the recipe for cupcakes, you need to have eggs and sugar and maybe some water and oil and some flour. And then let's say that you accidentally change this recipe so that instead of using sugar, you're now going to use organic sugar. Are your cupcakes going to taste any different? The answer is no. It's sugar. Whether it's just regular sugar or organic sugar, the substitution doesn't matter. You're not going to notice a difference in the end. Now, if it were a bad mutation, it would maybe mean that the sugar, instead of adding sugar, you added salt. Right? Now, that would be a bad mutation. You're going to end up with cupcakes that don't taste very good. But if the substitution is basically the same thing, or if it is the same thing, then who cares? Now, last but not least, but sometimes mutations are actually beneficial. So sometimes they're actually good. So it might be the sort of thing where the incorrect protein is made, but instead of making a worse protein, you actually end up making a better protein. So let's take a look at these insects here. So let's say that this resistance refers to being resistant to insecticide, like a bug spray. So if you have these bugs in your house and you spray it with Raid or some sort of insecticide, what's going to happen is that the individuals that don't have this beneficial mutation are going to die. But the individual that has this beneficial mutation is going to survive. Now, if this individual then goes off to make a whole bunch of little baby bugs, now all of these little baby bugs have that resistance built into their genes, and so they're going to be more likely to survive if they're exposed to that insecticide. So it sucks for us as humans because we have to keep making stronger and different insecticides in order to kill off these insects that we don't want, but it's good for the insect because it means that they survive and can reproduce. And this is actually the basis for evolution. So if the mutation results in some sort of beneficial trait that increases survival and reproductive output, it means that more individuals in the future generations are going to have that beneficial mutation. And then, of course, I had to put in the Teenage Mutant Ninja Turtles over here. Obviously, they're not real, but the mutant part of their name indicates that they have a mutation that allows them to, fight off criminals and eat pizza and live in the sewer.